Hello, today I'll present our work on incorporating contextual information into KGWAS for interpretable GWAS discovery. I'll start with introduction and I'll dive into our methods, experiments, and results. Let me start with the core motivation of our work. Human genetics and perturbation screens give us two different but highly complementary views of disease biology. Human genetics methods, such as GWAS, tell us which loci are associated with a phenotype, but this usually does not reveal the mechanism. It does not tell us the target gene, the relevant pathway, or the cellular mechanism. It also often requires large sample sizes to achieve statistical significance. On the other hand, perturbation screens provide experimental evidence for gene interactions and their downstream transcriptional effects in a specific cellular context. But they do not tell us which of these interactions or programs are actually relevant to the disease or the trait we care about. There is an opportunity to combine them using human genetics to anchor relevance to phenotype and perturbation data to supply mechanistic structure. One recent method in this space is KGWAS, which augments GWAS with large functional genomics knowledge graph. KGWAS aims to improve the statistical power of GWAS by representing variants, genes, and biological programs in a heterogeneous graph consisting of variant to gene, and gene to program edges. It then trains the disease-specific graph attention network to predict GWAS association statistics, improving statistical power in small cohort GWAS by prioritizing variants using their broader functional neighborhood on the graph. Importantly, the knowledge graph used in KGWAS reflects a broad view of universal cell biology rather than the specific cellular context of any one trait. This is because the edges are drawn from large general-purpose functional genomics resources designed to maximize coverage across many traits and tissues. For example, variant-to-gene edges are assembled from standard functional genomics mapping resources, including sources such as E2KL catalog, GTEx, ENCODE, as well as mapping strategies such as activity by contact and CS2G. Gene-to-gene edges come from broad interaction databases such as String, DIP, and BioGrid, and gene-to-program edges map gene to pathway annotations using gene ontology. This broad knowledge graph is the foundation that KGWAS uses to improve GWAS discovery and support downstream biological interpretation. However, the original knowledge graph contains substantial redundancy. Our work addressed this by making it more context-specific using perturbation-derived evidence. Before getting into our modifications, it's important to note that the original KGWAS framework already performed really well. Using the broad functional genomics knowledge graph, KGWAS showed that it can substantially improve GWAS discovery power in small cohort settings. In the original paper, across replication experiments on 21 UK biobank traits, KGWAS consistently identified more loci that replicated in the full cohort compared to the baseline methods, with the largest gains observed at the smallest sample sizes. The second important result is interpretability. KGWAS also uses the learned graph attention structure to derive disease critical networks. This provides the path from variant association to candidate mechanism in a way to generate hypotheses about how genetic associations may operate. As I mentioned earlier, the original KGWAS graph is broad by construction, since it is built from general-purpose functional genomics resources. While a universal graph makes the framework broadly applicable, it also means that for any specific trait, many of these edges may be redundant or lack specificity to the relevant cellular context. This redundancy can generate multiple plausible paths to represent the same underlying biological mechanism, ultimately reducing the generalization and consistency of disease-critical networks. Therefore, to make the graph more context-aware, we first sparsify it to reduce redundancy and then use the perturbation data to improve specificity by replacing broad generic gene-level relationship with cell-type-specific structure derived from the experimental data. Our goal is to test whether we can improve both statistical power and interpretability by incorporating perturbation data from trait-relevant cell types into KGWAS. Next, let me dive into the methods. At a high level, we keep the original KGWAS framework, then modify the knowledge graph to make it more context-aware. Relative to the original KGWAS graph, The first change we make is graph specification. The motivation is that the full knowledge graph contains overlapping structure, different subgraphs, and multiple edge types can carry similar biological information, and some edge types can be very broad or dominated by self-loops, which can dilute the message passing signal. Concretely, we first disentangle the contribution of gene-to-gene and gene-to-program subgraphs, and we decide to remove gene-to-program edges in our final model. We then prune low specificity edge types. For variant-to-gene links, we restricted to edge types with stronger functional evidence, including exon, promoter, EQTL, and FANMAP EQTL links, removing edges with broad mapping such as transcription start site proximity. For gene-to-gene links, we observed that many sparse relations are dominated by redundant self-loops introduced during graph construction, so we keep only the major gene-to-gene edge types with more than 10,000 connections, reducing the number of gene-to-gene relations from 46 to 12. We also evaluate collapsing edge types to test the importance of having these fine-grained relations. The second major change is to make the graph more context-aware using perturbation data. Rather than relying only on the original universal gene-to-gene structure, we define context-specific gene-to-gene relations using similarity in perturbational response profiles from a trait-relevant cell type. This yields a compact context-aware gene-to-gene graph with approximately 116,000 edges, defined directly from experimental evidence. Next, I'll dive into our experimental setting. To test our hypotheses, we use a publicly available genome-scale perturbacy screen in the K562 cell line by Rapogel and others. We apply standard perturbacy processing and compute log-fold changes for each feature gene. We apply independent component analysis and dimensionality reduction with 60 components, and then calculate cosine similarity between each pair of target genes, and use high similarity pairs to define context-specific gene-to-gene edges. To assess the biological signal in the dataset, we compare the pairwise cosine similarity for gene targets with known relationships documented in string. The relationships corresponding to string annotations have significantly higher cosine similarities, and based on this observation, we set a threshold of tau equals 0.5, yielding 116,000 high-confidence context-specific edges. We focus on 3 traits: MCH, IRF, and RDW. These 3 traits are previously selected by Ota and others in their work because the open chromosome regions in K562 exhibit significant heritability enrichment for these traits. We follow the training and evaluation protocol from the KGWAS repository. We train a 2-layer graph attention network for each trait to predict GWAS chi-square association statistics. For model evaluation, we subsample each trait to the same small cohort sizes used in the KGWAS paper, and we report the number of loci in the top 100 independent loci identified by each method that overlap with the ground truth loci discovered in the full UK Biobank cohort. Now let's dive into our results. I'll start by walking through the ablation studies performed on the original KGWAS graph, which together motivated the final design of our context-aware model. First, we asked which relation types in the original knowledge graph actually contributed to the performance. The key result is that removing gene-to-program edges have very little effect, which suggests that the gene-program relationships are not playing a major role in the default 2-layer KGWAS setting. In contrast, removing gene-to-gene edges causes a substantial drop in performance. We also repeated this experiment with 3-layer graph attention network, and there, the model becomes much more resilient by removing both gene-to-gene and gene-to-program edges. This suggests that in the original graph contains substantial redundancy because more message passing depth allow the model to recover these communications through alternative paths. Based on this, we keep the standard 2-layer architecture and remove the gene-to-program edges in the final context-aware model. Next, we examine whether the graph can be simplified to a more selective set of edge types without losing performance. Restricting to the higher confidence local variant-to-gene edges actually gives us a small improvement over the original KGWAS graph, and further pruning the gene-to-gene edges to only the major edge types does not hurt performance. Therefore, we can simplify the graph substantially without sacrificing recall, which is another strong evidence that the original graph contains a redundant structure. After edge type pruning, we collapse variant-to-gene edges into a single type and likewise collapse all gene-to-gene edges to a single type. Collapsing variant-to-gene edges does not materially change either the edge count or performance, which tells us that the most of the useful simplification on the variant-to-gene graph already come from selecting local high-confidence links. For gene-to-gene edges, however, collapsing remaining edge types further reduce the number of edges in the graph without degrading performance. So for the remainder of the study, we keep all local variant-to-gene edge types and collapse the major gene-to-gene edges into a single type. Finally, we evaluate whether perturb-seq-derived context-specific graph improves performance beyond our sparsified graph. Restricting the graph to the gene present in the perturb-seq screen already yields a more than 10% improvement, showing that context-specific gene prioritization is helpful. Adding, merging, or replacing the original gene-to-gene graph with the perturbation-derived gene-to-gene edge gives us a similarly strong performance. The replace setting is intriguing to us because it further sparsifies the graph by substituting the original graph structure with a much smaller perturbation-derived graph without sacrificing recall. So for that reason, we use the replace setting in our final model. The results in the previous figure were shown for a cohort size of 10,000. Here we show the full results with multiple cohort sizes, with the last row representing our final context-aware KGWAS model. The main pattern we see here is that the improvements are consistent across sample sizes, with larger gains appearing in the smaller cohort sizes where graph prior is the most valuable. At the same time, these gains come from a dramatic reduction in graph size. As we move from the original KGWAS graph to our final context-aware model, we see that the number of edges dropped from 12.1 million to 625,000, which is a 19-fold reduction. This shows that our context-aware graph is both substantially smaller and more powerful for these relevant traits. We perform ablation studies on our final context-aware KGWAS model to evaluate how much of the performance gain comes from the context-specific graph structure. Our results show that the full model performs the best, and the performance drops significantly when we randomize the perturbation-derived gene-to-gene edges, remove gene-to-gene layers, or randomize both gene-to-gene and variant-to-gene structure. This indicates that the improvement is not driven by a specification alone, but by preserving the meaningful biological structure that we have in the graph. One of our hypotheses is that making the graph more context-aware should improve not only the statistical power, but also the consistency of disease-critical networks. This is one of the key advantages of our method because it means the context-aware graph supports both better prediction and more stable biological interpretation. To evaluate that, we compare the disease-critical network produced by the original KGWAS model and the context-aware model across random seeds. In this example, the context-aware model produces a smaller and more reproducible network, where the original KGWAS network is less consistent across runs. To conclude, we show that the general-purpose knowledge graph used in KGWAS contains substantial redundancy, and that context-aware graph construction can improve both power and interpretability. Using perturbation data from trait-relevant cell type, we built a much smaller graph, about 19-fold smaller than the original KGWAS graph, while improving recall in a small cohort setting and producing more consistent disease-critical networks. As perturb-seq datasets become increasingly available, this framework can be extended to diverse tissues by matching disease traits to corresponding cell type-specific perturb-seq atlases, moving genetic discovery toward a more context-driven paradigm. Finally, I would like to thank all of my co-authors for their contributions to this work, and thank you for watching.